Welcome back, guys, to our second session of our algal bloom exploration. In this session, we're going to start off with a reading on algal blooms to get a little bit more data on it, and then we're going to engage in an analysis of an experimental design where we will be identifying the independent, dependent, and constant variables. So what happens during an algal bloom? Algae are plant-like organisms that live in water. Algae are able to capture sunlight to make sugar during photosynthesis. Algae can come in many forms and can be grouped by how many cells these photosynthetic organisms have. Single-cell organisms have only one cell and rely on their cell structures to carry out life functions. Multicellular algae, like seaweed, have their millions of cells organized into tissues, organs, and organ systems. When the environment changes so that there is more nutrients, single-celled algae multiply out of control. As the population explodes, there is so many in a given area that they start to appear as a colored film in water. So in this experiment, Roman wants to study the effect sunlight has on algal growth. He adds nutrients to both beakers, but places one next to a window and the other in a dark closet. All right, so here are how the beakers are set up. You have beaker A next to the window and beaker B, which will be placed in a closet. Beaker A has 250 mLs of tap water. We also added 2 mLs of cyanobacteria. We set it up in a sunny window for 2 days, and we added 1 tablespoon of fertilizer and mix. Now for beaker B, we added 250 mLs of tap water. We added 2 mLs of cyanobacteria. We set it up in a dark closet for 2 days, and we added 1 tablespoon of fertilizer and mix. So now I want you to brainstorm what you believe will happen to each beaker after the 2 days. When you're done thinking about it, you can go ahead, press play so we can continue with the work. So here we are. These are our samples after 2 days. This is sample A after 2 days and sample B after 2 days. And you have a magnified view of both samples underneath so you can observe the amount of algae growth. So you can take this opportunity to press pause and make some careful observations of each beaker. So one of the main things you may have noticed is that beaker A had a lot more algae growth than beaker B. So I want you to think about what could have caused the different results, right? And that brings us to our variables. So here we have 2 different variables. We have our independent and we have our dependent variables. I've also included constants for this so we can continue to get review on those. So for independent, I always refer to it as the cause because that's really what it is. What caused the different results, right? What did we change that caused the different results? And you can use this prompt to help you. What I changed was blank. For the dependent variable, that's the effect. That's the outcome or the results, right? So usually I refer to it as the data, all right? So once again, cause, independent variable, and effect, dependent variable. I've also added constants, which are things in our experiment that stayed the same in order for the experiment to be fair. So before we go ahead and have some time to determine the cause or the independent and the effect, the dependent in this lab or experiment, I am going to show you a video that recaps variables quickly in case you need a little bit more of a review on them.
There are 2 main types of variables in an experiment, the independent variable and the dependent variable. The independent variable is the difference between the control and experimental groups of an experiment. The independent variable is the variable changed by the scientists to test the experiment's hypothesis. The dependent variable is the thing being measured during an experiment, also called data.
All right, guys, now that we had that quick little recap, go ahead, press pause, and work on the independent, dependent, and constants for our experiment. And when you're done, press play so we can review the responses. All right, guys, welcome back. And now let's check in and see if you were on the right track for identifying your variables. So, what is the cause? What do we change here on purpose? If we go back to our setup, you'll notice that one beaker was exposed to sunlight, but the other was not. It was actually in the dark. And we know that one of the main requirements for algal bloom is sunlight exposure. So the independent variable is sunlight exposure, right? How much sunlight was the beaker exposed to? Now, what is the dependent variable or the results, right? So we look back at our data and the results were the amount of algae that grew, okay? So that is our dependent or our effect, how much algae grew. And last but not least, let's look at our constants. What are some things that we kept the same throughout the entire experiment? So some constants include the amount of tap water, which was 250 mLs for both beakers. Also, the amount of cyanobacteria and the amount of fertilizer. Each beaker received 1 tablespoon of fertilizer mix. All right, guys, so this marks the end of our review. I wish you the best of luck in your independent work, and I hope to see you again for future videos. Take care and bye-bye.